Sleep originated in very early animals and is seen all over the animal kingdom today. When we think of sleep, we usually think of the brain because the nervous system generates sleep, we dream during this time, and overwhelming mental fog follows even a single night of sleep restriction. But can sleep really be all about the brain? The fact is that many diseases which have no obvious connections to the nervous system can be made worse or even be caused by poor sleep. Severe sleep restriction can even lead to death. This has been shown in various model organisms. So, to understand the vital nature of sleep, we asked, what do sleep-deprived animals die from?
To answer this question, we first used fruit flies whose sleep is similar to our own. We kept them awake for many days, which led to premature death. So, what killed these animals? The key here was to be unbiased and examine various organs at the same time for signs of cellular damage starting early in the deprivation. And we found that severe sleep loss causes accumulation of reactive oxygen species, or ROS, and that this happens specifically in the gut. The gut on the left is from a fly that was sleep deprived for 1 day, and the one on the right is from a fly that was sleep deprived for 10 days. These guts were stained with a dye whose fluorescence changes when oxidized by ROS. So here, a brighter fluorescent signal indicates higher levels of ROS. And as you can see, ROS levels are significantly higher in the gut of the fly that was sleep deprived for longer. ROS accumulation leads to oxidative stress and cellular damage in the gut. But when the deprivation is stopped and animals are allowed to sleep, ROS levels gradually decrease and the damage is cleared. So we then ask if this phenomenon is conserved in mammals by examining sleep-deprived mice. And just like in the fly, sleep deprivation leads to gut-specific ROS accumulation, which triggers oxidative stress in this organ. The small intestine cross-section on the left is from a mouse that was sleep-deprived for 1 day, and the one on the right is from a mouse sleep-deprived for 5 days. And as you can see, ROS levels are significantly higher in the mouse intestine after longer sleep restriction.
So, there is a strong correlation between sleep loss and ROS accumulation in the gut. But is this why sleep deprivation can be lethal? To test causality, we returned to flies and asked if inhibiting ROS accumulation in the gut prevents death during severe sleep restriction. We were able to get rid of ROS with either oral antioxidant compounds or by overexpressing antioxidant enzymes in the gut. In both cases, normal lifespan is restored, while the animals remain nearly sleepless. But what about the nervous system? Well, we could not detect any obvious signs of ROS accumulation in the brain in either flies or mice, and overexpressing antioxidant enzymes in neurons does not prevent premature death of sleep-deprived flies. These survival curves show the premature death of sleep-deprived flies in red, compared to the non-deprived controls in gray. The brown curve represents the survival of sleep-deprived flies that overexpress an antioxidant enzyme, SOD1, in their gut. Preventing ROS accumulation in the gut of these severely sleep-deprived flies allows them to live as long as the non-deprived controls.
Our study identified intestinal ROS as a major cause of death in sleep-deprived animals. In the paper, we discuss what we think this means in terms of sleep origin and function, and how intestinal ROS could explain the link between poor sleep and poor health.